Good afternoon, everybody. Thank you for making me a part of this. Um, so I am a genetic counselor, and my love is genetics. So ataxia, so any disorders that have a genetic background, I kind of want to know more about it and want to see how I can help as a geneticist in that field. And this opportunity was given to me by Ms. Swasti Wagh, and I'm very humbled and honored to be called here today. So we touched upon a little bit about genetics of ataxia. Before we go into that, I want to talk about what is genetics. I know here there are many students who are masters and there are patients, and we have heard the term genetics a lot. But what is it? What is genetics? See, what is happening is our body is made up of cells, right? Uh, it's like a building block. Every cell has a DNA, right? So here we see there is the DNA, which is made of small nucleotides. So a string of nucleotides is called a DNA. Now this DNA is packed in a chromosome. Okay, so I'm basically starting at the very basics. I know we all know what it is, but if there is even one person who has forgotten, like I have forgotten by my masters, I have forgotten what I learned in 12th standard. So basically, string of nucleotides is DNA. Now this DNA is packed. How many of us have gone to Toshiba and bought a NADA, right? We buy it by meters, but how do we store it? We coil it around and we make a small package and then we store it. Similarly, this long string of DNA is coiled around and packaged as a chromosome, right? Important part here is every cell in our body has the same DNA, has the same number of chromosomes, has the same genomic DNA. Okay. However, one part develops as a hand. As an embryo, think about it. All the cells have the same DNA. One part develops as hand, one as nose, one as foot, because the regulation is different. At what time, what part of the DNA will be active is decided by the regulatory system. Okay, these are just the basic things which will help us understand the disease. Okay, so there are two things. Our genomic DNA is made of one, that is the coding region with the part that forms the proteins. What time which proteins should be formed is controlled by the second section of our genomic DNA, that is the regulatory section. This may microRNAs, small nucleolar RNAs, promoter regions, and epigenetics is all covers that. We'll not. And so basically, one more point before we go into details is how many chromosomes we have. अभी तो मैंने answer पहले ही बता दिया. But how many remember how many chromosomes we have? किसी का भी हाथ ऊपर नहीं है, so I'm assuming nobody remembered. It is just me who's telling. So we have 46 chromosomes, 22 somatic pairs. When we say 22 somatic pairs, meaning every individual, male or female or other, they have these 22 somatic chromosomes. Two sex chromosomes. In females, we have two X. In males, we have one X and one Y. This is the normal karyotype. Okay, so my PhD was a little bit in molecular evolution, and Dr. Ichaporia also touched upon evolution. So this is a very big evolutionary advantage that we have every gene in our body as a copy of two. So if something happens to one gene, we have another gene to kind of compensate for it. If you look at hemoglobinopathies, we have multiple copies of genes for alpha hemoglobin and all. Why? This is just an evolutionary mechanism for us to protect against कि एक mutation हो गया and the person dies. That doesn't happen. We will have compensatory mechanisms, especially when there are important functions like blood, like hemoglobin. When it is that important, then there are many, many, many copies. So सिर्फ दो नहीं है, और भी duplicates बन जाते हैं. So this is the body's like nature's way of kind of protecting us. How does it do that? We'll talk about it. So now what is ataxia? Genetics part over, ataxia started. So I am very glad that both the speakers before me explained it very well, and I don't need to do it. I was a little bit scared because this is a new topic for me. But basically, what I understood is ataxia is like an umbrella. Under that umbrella, any disorder that causes the gait problems or the coordination problems, any disorder that causes these problems, these symptoms, they fall under the umbrella of ataxia. I think I'm correct. So basically, what are the symptoms? Poor coordination, walking unsteadily. Madam displayed it very well in the videos also. Poor balance is there, difficulty fine motor speech, motor skills, change of speech, involuntary movement of the eye. Sir talked about it, and difficulty swallowing. And these are just the symptoms. And both sir and madam highlighted how we could take care of that. So, from my point of view, from a geneticist point of view, I will say there are two types of ataxia. One is acquired and one is hereditary, right? Acquired, may madam had given many classifications, but acquired is head injury that sir also talked about. Any infection, bacterial infection, viral infection, cerebral palsy, multiple sclerosis, alcohol misuse, thyroid gland, B12 deficiency, sir talked about. Now, all of these ataxias, they need to be, I think a doctor needs to see them and classify them and treat them accordingly. Basically, is whenever there is a problem with the cerebellum, anything that blocks the cerebellum function, we can develop ataxia, right? However, this is acquired ataxia. And what is the hereditary ataxia? Hereditary ataxia meaning there is no external cause and still a person is not able to perform the day-to-day activities as easily as somebody else of that same age can do it. In children, in adults, there are adult onset ones, there are ones that are in manifest much earlier in the time. So how do we diagnose hereditary ataxia? Family history is very important. We have to go to a doctor, maybe a genetic counselor, but that comes later. Pehle to the patient will come to a hospital only. Maybe they will come to an orthopedic person, doctor first. Then the orthopedic will say, no, 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 go to neurology. Then they will go to neurology. Then from neurology, they will tell all of these things. And so at the first level only, the family history needs to be taken. Physical examination the doctor will perform to kind of identify if it is ataxia. Neuroimaging, sir talked about it. And then what happens last is molecular genetics. There are many gene panels that are available that will check for mutations in any of the ataxia genes. I'll show you the long list of genes, but before that, a little bit background I will give. So age of onset, sometimes after the age of 50 it shows, which can be still hereditary. Don't think that hereditary means as soon as the baby is born, they are showing symptoms. It will be after, these are called late onset disorders. Around the age of 20, before the age of 20, Friedreich ataxia is there or ataxia with oculomotor type 1 and type 2 apraxia, type 1 and type 2. And before the age of 5, as a child, as a little child, you see ataxia telangiectasia, which is manifest as an infant also. I think hypotonia must be there and some other symptoms can be there. So basically, age of onset, anyways, hereditary. अगर late भी है तो भी think about it that it could be hereditary. One thing that we have observed is that sometimes a baby is born and they are showing ataxia, and then we say, oh, ye kaise aa gaya? It was not in the family. However, what I feel, what we have seen is that sometimes the parent had the mutation, but it never manifested. But because of anticipation, when a mutation is passed to the next generation, uski severity बढ़ जाती. That is called anticipation in a genetic counseling term. So because of that, sometimes we will see it in the child, but we will not think of checking the parent. So if a child is there and the parent, we have not checked if the parent has the mutation or not. Maybe it is important to check the parent also so that when they are having a second child, the risk we can counsel them. And there are tests that can happen with CVS, with amniotic fluid, where we can identify if the second child will also be having this condition or not. So that is very beneficial to families because as hereditary, we may have more than one child with the condition, and then it becomes very financially and mentally also very challenging for the family and even for the siblings themselves. Uh, so, so then there is the episodic ataxia. I'm admitting everywhere, even here. So episodic ataxia is there. So sometimes ataxia are, if you see a patient where the ataxia is associated with other disorders like retinopathy, seizure, dementia, just dizziness and vertigo, muscle cramping, scoliosis. So here, you remember Sir talked about SCA, it is spinocerebellar ataxia, and there were 1 to 49 he had uh, written here on the screen 1 to 49, and each number denotes the characteristics of that type of ataxia. Okay, so now these are the characteristics where if the ataxia is with retinopathy, it could be SCA7. If ataxia is with seizure disorder, it could be SCA10. Infantile onset, SCA7. So now we sort talked about autosomal dominant, autosomal recessive, X-linked, and mitochondrial ataxia. So now I just want to give a little bit brief about what these terms mean and then what are the mutations that will pass on in this fashion. So we'll start with autosomal dominant. I hope we can see it. Yeah. So basically in autosomal dominant, so this I talked about, everybody had two pairs, like a pair of per chromosome, there is a pair. So there are two copies, right? Two copies in the father, two copies in the mother. Now, father has one affected copy, one mutated copy, right? The view here shows the mutated gene, and this is autosomal dominant. So, although the father has just one copy, he will still be affected, okay? But it could be a late onset. And then they have children. So because it is autosomal dominant, whichever child will get that one copy of chromosome from the father, the affected mutated gene from the father will develop the symptoms. So 50% chance that a child born to this couple will have the mutated gene and show symptoms. So what are the genes in ataxia? This is clear to everybody, right? How this is inherited? So what is the autosomal dominant? So it is very slow progressive and it is associated with cerebellar atrophy, right? So Sir had shown pictures depicting cerebellar atrophy. So that can be known from the MRI. So now we have about SCA1 to 15 on this page and there are more on the other page. The gene associated with each disease and the average onset of these genes and what is the duration and the distinguishing features. So ataxia SCA1 to 49 here, and it is not just one gene affected. Different genes can give this symptom, right? Here. So we see here SCA1 may, if I am suspecting, I look at the features and I'm suspecting that this patient may have SCA1, I will ask for this gene to be tested. Okay. So then, so basically it becomes a little bit difficult for the doctor because if you read this, many may be overlapping. So that's why we have the gene panels. All these genes will be put in one panel. We can go to one of these companies that do this genetic diagnostics using next gen sequencing. We tell them the whole panel. So basically, the panel is about 250 plus genes, 57 or something like that. So that is the panel. And in that panel, all these genes are checked and they will identify the mutation, right? And once the mutation is identified, it becomes very easy for a counselor to counsel the patient on what is the chance that the sibling will have this risk or moving on to the next generation. So these are next set of autosomal dominant genes that are, yeah, these are next sets here that are affected, and this is HCA 17, 18, 19, 22. So the KCND3 gene is uh, affect is causing symptoms for both 19 and 22. So now you have to remember that genetics आने से पहले genetics का study आने से पहले these classifications were given. So 19 and 22 दोनों के symptoms can be seen if this gene is mutated. Now why is that? It depends on where the gene is mutated. उस जो भी प्रोटीन बन रहा था उसका कौन सा फंक्शन अफेक्ट हुआ है, which domain of the protein is affected. So it will be a deep molecular biology study to check that, right? And age of onset, if you see, fourth decade, adolescence, fourth decade, 6 to 30 years. So this is so varied, yeah. So that is also something that we need to uh, once we are counseling a patient with ataxia, we have to see कितने साल में आया है and all that, and the kids we have to check for because they may be more severe. So some more genes and some different types of ataxia besides ACA, SCA, there are different ones which may also be caused by genetic inheritance. Now, autosomal recessive inheritance, just like autosomal dominant, we see if just one parent had it, and if it was just one carrier father, for example, so only 25, not even 25. So none of the kids will have that, will show any symptoms. However, if a carrier father and a carrier mother marry and they have kids, then there is a 25% chance of one of the child being affected. And two children will be carriers and one will be normal who is not carrying any mutation. When we are counseling a patient like this, we have to see consanguinity because consanguinity is the biggest problem where this arises because that mutation is segregating in their family, and then somebody in the family marries the other. There's a higher chance that the every person in that family is a carrier. So it is better to check while taking family history if the patient is a carrier, like consanguineous marriage is there. So these are some very common ataxias, but which are uh, autosomal recessive. So Friedreich ataxia, ataxia telangiectasia. So basically, this is at onset is first decade, 10 to 20 years, but it is much earlier also, and it is associated with cancer also. So that is something that we need to counsel the patient and have regular checks. And then different types of ataxia. These are all the mutations that are inherited in our autosomal recessive family. Another type of one is X-linked inheritance, inherited where normally the males are affected because X-linked matlab that mutation is present in a gene that is on the X chromosome. Okay, so X chromosome from the mother and the X chromosome from the father, they get together to make the female. And if it is recessive, X-linked recessive, then it will not be expressed. But if it is X-linked dominant, it will be, most of the time it is X-linked recessive. And so from the mother is a carrier in this case, and she has given it to the son and the daughter. The daughter, she will not be symptomatic because if it is recessive, if it is X-linked dominant, then she will be also symptomatic. But the son most definitely will be symptomatic, even if it is recessive, because son has only one copy of X, right? So So what happens in X-linked inheritance? It is mostly early onset ataxia. These difficult words are there, so just pronounce it. Dyssynergia and dysdiadochokinesis. We have been explaining these both terms very nicely, so I'll not even try to go there. It is either non-progressive or very slow progressive. Uh, so there are like in some males, so this is mostly in males if it is X-linked. Upper motor neuron signs will be there, are present in some of the males. Mild learning disability is seen, anemia is mild. Carrier females have normal neurological examination and the mutation is in ABCC7 gene. So females that have this is normal, and sometimes it can be uh, early onset ataxia, and it may be a part of the fragile X. Sometimes these mutations, so fragile X syndrome is also an X-linked disorder that mostly the men have, and this ataxia can be the symptoms may be shared between fragile X and mitochondrial inheritance. So now, just think about embryology. There is the egg and there is the sperm. So the sperm does not carry mitochondria. Only the egg carries mitochondria. So only the maternal, if the mother is having, if it is a mitochondrial mutation, only it will pass from mother to every child. Not, it does not differentiate between male or female, right? So mother will transport to every child. The daughters will pass it on to all her children, but the sons will not, because the sperms will not carry the mitochondrial DNA, right? So in ataxia, how does it matter? So it is a progressive form of ataxia, and they're associated with additional clinical manifestations just like seizures, deafness, diabetes, and it is basically a missense mutation in this gene, MTA-TB6. And it can be both childhood and adult. So now again, uh, this becomes very difficult for a doctor to diagnose. So that's why the family history becomes so important. And then the gene panels, we have to get tested. So these are the genes that are present in. So no, this is one thing that I wanted to leave you with. Sir also talked about it. So now the understanding that every mutation is like hereditary ataxia cannot be cured as a nahi hai. Even hereditary can be. There are a little subset. It's a small subset, but it is there that is potentially treatable. That is why it becomes very important for us to at least find out what is the mutation, where is the mutation, if it is caused by a mutation. If it is not acquired, then it becomes a duty for us to kind of check what is the mutation, and then if it is treatable, then the patient will benefit a lot. Like if we can treat the patient by vitamin E supplement or replacement therapy. So if the mutations are in these genes, there is a chance that we can maybe reverse it. And also these are some other one, which is coenzyme Q10 deficiency. And we can give a coenzyme Q10 replacement therapy. And there's a possibility that if it is mutations in these genes, it can be reversed. There's a possibility that it can be reversed by coenzyme Q10 replacement. So that is all I have. I'm not going to talk a lot more. I don't know how much time I have taken, but basically, take-home points are ataxia, two types, acquired and hereditary. If anybody is suspected of having the hereditary ataxia, get the gene panel done. There are many companies. It is a little bit expensive, but sometimes for the poor patient, if you can provide the proof that they are under BPL and something, most of the companies they give discounts. If a patient can afford, definitely you should go for the gene panel. The ataxia gene panel is there. Now, I'm from MUHS. I just wanted to talk a little bit about Maharashtra University of Health Sciences. We have started a lab here called MUHS Gene Health, and we are providing genetic counseling for the poor patient for a mere 10 rupees. So because we have counseled about 9, 10 patients and we forgot to take that 10 rupees also from them. So we, we are basically counseling for free because it is also a training center. So for our students, it's helpful if the patients come and we can counsel them and the students can also. So, if there are any patients that you think need genetic counseling, please connect with me. We have experts that are on our panel, and we can call those experts, which are specific for neuromotor disorders or specific for pediatric or any other. We just pull them together and we'll call the expert and we'll plan them. That is one. Second thing, actually, Ms. Wag was very much insistent that we should start some research in ataxia here, genetic research. So I please connect with me. We can think of projects to write. MUHS does provide some funding, some basic seed funding. We can see if we can be eligible for that or not. So we have the tools. We don't have NGS. We have basic molecular biology. The whole setup is there. For NGS and all, we are trying to collaborate with others. We have an MOU with NARI. We have MOU with NCCS. So if there is a bigger project, we can write it with them. So do connect with me, and we can think about research if we can do it at our, because funding is the biggest issue. But because MUHS has agreed to give basic seed fund, we can think if we are eligible under this topic. With that, I will say thank you. If there are any questions.